All right, so after that sort of very quick overview of probabilistic modeling, Bayesian inference, et cetera, let's turn to something a bit more applied and see how we can use some of these ideas to build, hopefully, what is an interesting model. So with some apologies, I looked over the papers you presented in the last couple of days. I realized PyClone and DDClone were in there, so this is going to look remarkably similar. It's a bit boring maybe, but the point here is not the model. I'll reiterate that. It's the process that we're going to go through, right? So try to focus on that aspect here. There we go. All right. The good news is I can blow through these slides pretty quickly because hopefully you've seen this. The basic problem we have here is that there's a clonal population structure in cancer, right? So at the time of biopsy, we have all these different populations. Now, when we do high throughput sequencing, the basic idea is that, or at least the basic idea of this problem, is that we have a heterogeneous set of cancer cells with different mutations, different stars, and when we sequence them and look at our short read data, the number of reads with a mutation is some way dependent on how common that mutation was in the population we put in for sequencing, right? Now, just a bit of notation. The way we summarize this, before I blow through this slide, this is obvious to everybody, or is no, not okay. So maybe I won't go through so fast. Okay, so the setup here is you have high throughput sequencing. You have input cells. In order to do high throughput sequencing, you break the cells apart, right? And essentially throw them in a blender, release the DNA fragments, and then you get your short reads out of sequencing that. You take your short reads and you align them back to the reference genome, and that's what this picture is showing here, right? And essentially what we're trying to show sort of qualitatively is if you look at the purple mutation, which is relatively rare in our input population, there's fewer reads that actually support that mutation. Now, there's a bit of notation here. Dealing with sort of aligned reads is not the easiest thing to build a model with. So what you want to do is summarize that down to something a bit simpler. And the basic quantities we use is this vector A of reads that match the reference, right? And B, reads that are a mismatch to the reference, right? So for example, we have 2 reads with the mutation, so B is 2. And then we have the total depth of coverage. A little bit of terminology. We'll talk about the variant allele frequency. That's just the proportion of reads with the mutation. And there's the cellular prevalence, or cancer cell fraction is another name for this, and that's just the proportion of cancer cells with the mutation.
Is that cancer cells or cancer with the mutation?
Pardon me?
Because it says proportion of cancer cells and then with the mutation, is that the ones carrying this specific variant?
Yeah.
It's involving cancer cells. It's not just a mixture of normal with cancer.
No, so the proportion of cancer cells overall is, we'll call the tumor content.
Okay.
This is, if we think about just one mutation, what proportion of cancer cells have that? We'll get back to that in another slide too. So the high-level question here then, given that sort of setup, is given bulk sequence data, can we estimate how common a mutation is, right, in the input? And can we also potentially group mutations together to infer which ones coexist within the genome of clones, right? So can we start to work out the genotypes of the clones, right? So there's a really simple solution that has been applied. Here we see our heterogeneous clonal cell population. We've sequenced them. And what we can do is plot the variant allele frequency. So this has been done on 2 axes with primary and a relapse tumor, right? So for example, mutations down here are only in the primary and not in the relapse. They're at 0 variant allele frequency, right? And if you plot the variant allele frequencies of the mutations, you start to see this clustering pattern, right? So there are groups of different mutations. No questions? And in fact, that's sort of how they built this picture, right? So this sort of makes sense. Maybe we should just cluster mutations. But there's a bit of an issue here. And before I talk about it, I just have a slightly technical point. Should we even be looking at the variant allele frequency for this problem? Remember that we're measuring two independent things, the number of reads with the reference and the number of reads with the variant in this problem. But when we compute the variant allele frequency, we end up with one quantity, right? So maybe the bias in my voice is giving this away. Is it better to use the variant allele frequency or the allele counts? So maybe this will help. What's the variant allele frequency? Pardon me? Oh, you can't read that? Here, 1 second. I'll do this in a much larger. Is that any better? Still too small?
Yeah, it's bigger.
Okay. A is 10, B is 10. What's the value? Half, right? Yeah, it's 10 over 20, 10 plus 10. Let's do this. Okay, a little bigger this time. What's the value? Right. Okay. What do we lose when we use variant allele frequency? The absolute counts, right? And remember, this is just an estimate of the proportion of reads with the mutation, right? Which of these two is going to be a more confident estimate? Right, we have more data, and if we see it 1,000 and 1,000, we're even that much more confident, right? So the problem here is if you convert to variant allele frequency, you're throwing information away, right? And that information is really your confidence in the measurement. So, go ahead.
I guess, like, my question is I'm sorry I don't have a lot of background for this, but I don't really understand what's the difference between the reference V and the variant.
Okay. Cool. I'm going to try to get to a less squeaky place here. Go ahead.
I don't really, like, I want to follow the pipeline, and I don't really understand.
Yeah, so the reference allele is the thing we see in the human reference genome, right? The variant allele is anything that's different, right? So if we have a point mutation, we're going to change to something different. Now, the reason we don't try to actually say is it an ACGT is because we're assuming that we never get a point mutation twice at the same position. So we may see, for example, the reference is an A, and we may see a C, right? That's our mutation. What I'm saying, I don't also expect to see some reads with a T, because the genome is really big, mutation rate is not that high, this position should not get mutated twice, okay? That's the assumption. And that's why we don't actually differentiate all of the counts for the different ACGTs. That's why we just say reference variant. This breaks down sometimes, at hotspot particularly. In general, though, it's not a terrible assumption. You can debate that if you feel strongly, but it's a simplifying assumption anyways. Does that clarify?
Yes.
Good. This is a technical point, maybe for the people who do modeling. what's an appropriate distribution to model variant allele frequency? Just shout something, please.
Two-point distribution.
For the variant allele frequency?
Oh, sorry, no.
Okay.
Beta?
Beta's beta's the,
Is it between 0 and 1?
exactly. The variant allele frequency is a continuous value between 0 and 1. Beta is a good one, right? You will also see people use the normal distribution. That is not a great choice because it's a distribution with support from negative infinity to infinity. It works pretty well, but it's not right. And what about for the count data, for the allele count data?
Binomial?
Yeah, so if we condition on the total depth, binomial, so we could model the counts of the variant as binomial. Or if we treat them independently, we could model them as a Poisson, right? Let's see if I can get my pointer back. Here we go. All right. So that was a little bit of a digression about data representation. Let's go back to this idea of clustering variant allele frequencies for a second. So one issue you run into pretty quickly, I'm losing my pointer, you run into pretty quickly is that the variant allele frequency is not an accurate estimator of how common the mutations are. So I'll keep going here. So the variant allele frequency is not the cellular prevalence. This is sort of a cartoon to illustrate why. If you have diploid mutations, so here we have a star, which is the mutation. We have 2 chromosomes. One of them is mutated. All of the cells have the mutation, but when we sequence it, only 50% of the reads are the chromosomes we would expect to show the mutation. Is that clear to everybody? So if all our cells are diploid heterozygous cells, what we can do then is double the variant allele frequency to estimate the cellular prevalence, right? Making a lot of other assumptions, but in this simple model where we have a homogeneous population of diploid heterozygous mutations, right? So that's how we could use the variant allele frequency. But that gets a little bit debatable, right? So there's some big assumptions in doing that. So here's a figure I've taken from an older paper. I think this is lung cancer. And so they're basically looking at variant allele frequencies from a sample. And they're looking at density plots. So this is the variant allele frequency here. And this is one patient, we see a single mode. In another patient, we see 2 modes, right? And so they're saying here we have a monoclonal, and here we have a biclonal distribution, right? And it's a little hard, I don't think you can read this, but this peak is basically at like a frequency of 10%, and this peak is at a frequency of 20%, right? So do you guys buy that? Is this a reasonable inference that this 2 peaks is biclonal?
Can you repeat the question, please?
Yeah. So if we were to see this 2-peak distribution of variant allele frequencies, is it reasonable to say we have 2 clones? Is it? Could come from
the same clone. So both mutations or both variant frequency can come from the same clone.
But why would they be different? Why would we get 2 peaks though?
Well, because each one of them has a different variant frequency.
But why the variant allele frequency? I think you're on the right track, but why?
Because a different number of reads allow to the different mutations.
So this is over quite a lot of mutations. So that could be a stochastic effect, but over a lot of mutations, we're seeing a bunch that are at 10% and a bunch that are at 20%, right? It's not just one read supporting this. This is, say, hundreds of thousands. So it can't just be sort of a random chance event, but that, I mean, that would be an explanation, but there's lots here. Okay. Are all mutations heterozygous diploid? No? No, they're not. Cancer genomes are genomically unstable, right? So if they were stable, then all mutations would likely be heterozygous and diploid, right? They'd only happen once at a location, then nothing else happens, right? But as soon as you have genomic instability, the picture changes. We can get things like this where both copies are mutated. It's important to emphasize here, we're not saying that we have two independent point mutations here. What the explanation is that we've lost one copy and then we've had a duplication at the same locus, right? This happens all the time. This is copy neutral LOH. We can also have an amplification of one of the chromosomes. Now we have 2 and 3. We can have balanced amplifications through genome doubling, et cetera, right? All of these will lead to different variant allele frequencies. So a very plausible way, if we go back to this picture, to explain these two peaks, half of those mutations look like this, half of those mutations look like that. We'll come to the sort of math of that, but essentially, going back all the way to this picture, here, because they're diploid, we have 50% with mutation. What happens if both alleles had the mutation? What would be the VAF? 1. Be double, right? Because all of them. That's why 2 peaks is maybe not the most parsimonious, 2 clones is not the most parsimonious answer here. All right, this brings us to a concept we'll call mutational genotype, and we're going to think through a very simple model for this. So the goal in this section, and just to put this in context, eventually we're going to arrive at a model that looks like PyClone for those who read the paper. But we're not going to go all the way for that initially. We're going to start with a much simpler problem that we can look at, and then we're going to build on it. And so the goal of this section is going to be to find a model which relates our count data. Here we're just going to use the number of reads with the variant, and then the total number of reads. And we're going to try to relate that to the cellular prevalence, this latent variable or hidden thing that we want to measure. To do so, what we have to think through is how is our data generated, right? And what we'll do is do a very common, use a very common technique in probabilistic modeling, which is to introduce something that we want to know and just assume we know it, and then think through what the probabilities look like. And then we basically use the machinery of Bayesian inference to figure out how to learn that. So the questions that we'll go through here is, how does the data relate to this latent parameter, which for us, again, is the cellular prevalence, how common the mutation is in the cancer cell population? What is the model that generates the data given the cellular prevalence? This is where we think, if we knew this, how could we how could we simulate the data? And what assumptions are we going to need to be able to actually make eventually to evaluate this probability? So the way we're going to proceed is by thinking about one mutation at a time. So we're just going to focus on a single mutation out of the thousands that the cancer cells have. Now, with respect to this one mutation, we can break the cell population into 3 parts. Can everyone still hear me over that? Solution is to move closer if you can't. We're going to break the population into 3 parts. So there's going to be the normal population. This is the cells that are not malignant. So in any tumor, there'll be some proportion of cells, either immune or just surrounding stromal tissue, right? Then we're going to have the reference population. So these are cancer cells that do not have the mutation. And then there's the variant population. These are cancer cells that do have the mutation. So I'm going to say this one more time because people tend to get confused on this point. This is all with respect to a single mutation. So the question I get asked quite a lot is, how do you have cancer cells without mutations? You do not have cancer cells without mutations. They do not have the one mutation that we are thinking of at this point. They may have a bunch of other ones, but we're ignoring those, right? If we were to look at a completely different mutation, then these cells might shift around into which some will become be in the variant population, some will move to the reference, right? It really depends on the specific mutation we're looking at, okay? Is that crystal clear? 'Cause if you don't get that, everything else stops making sense from here on in. Okay, and then the basic problem becomes figuring out how big the variant population is out of all of the cancer cells, right? So we're going to have to simplify that picture a bit, even though it's already pretty simple. In that picture, the mutational genotype, and by that I mean the combination of the number of chromosomes and the number that are mutated, that is the mutational genotype. In the picture, every cell has a different mutational genotype, right? To write down a probability of the data, we would then need to know the mutational genotype of every cell. That's seems pretty intractable. So what we're going to do is make a pretty dumb but useful simplifying assumption. We're going to assume that the genotype, mutational genotype of every cell within a population is the same. So the mutational genotype is now the same for all the cells in the variant population. It's now the same for all the cells in the reference population. Does that make sense? No? Okay. This is an assumption. It's definitely violated in real data. If you don't make it, it's quite hard to build a model though, right? So here we see the compromise of having to make a simplifying assumption that we know maybe doesn't reflect the real world problem well, but it does give us something tractable. What we hope is that this is not massively violated in our data sets, right? And really what that amounts to saying is that essentially most of the variant population is coming from one clone defined both in terms of copy number and point mutation. So that picture seems maybe a bit useless, but it does allow us to pose the following question. What is the probability of sampling a read with the mutation? So given our input set of cells, what is the probability of sampling a read with the mutation? We're going to have to imagine the process that generates our data now, right? So, and this is sort of what I explained earlier when I had the count data up. We start with a tumor, we take a sample. Now we take that sample and we essentially put it in a blender and break the cells apart to release the DNA fragments. That creates a pool of DNA fragments. We're going to assume that that pool is infinitely large. That's a technical point, and we'll come back to that. But there's just sort of, imagine a soup of DNA fragments floating around. And what we're going to do is we're just going to pull one of those out and sequence it. And then the question is, what is the probability that had the mutation? So does anyone know why we're going to assume that pool is infinite?
Otherwise, I just switch on you to get a sample, so it changes the
Independent row.
Pardon me?
Independent row for the infinite population, you don't have to account for the...
Yeah. Yeah, so you guys are both saying exactly the same thing. Essentially, if we don't make the assumption it's infinite, then once we remove a fragment, the probability of the next one changes because the number of fragments that had the mutation is different, right? So this is a classic sort of simplifying assumption one has to make. Well, doesn't have to, but often does make. So now the question is, how does this help us compute this probability? And the basic strategy is to break the computation into two parts. We're first going to assign the fragment to a cell population. So we're going to think, we pull the fragment, and the first thing we're going to say is, now we know which population it came from. Normal, reference, or variant. Right? Not clonal population, but populations as defined earlier, normal, reference, or variant. And then, given that the fragment comes from that population, and here's the key insight. Once we know which population the fragment comes from, then it's very easy to compute the probability that fragment has the variant, because we know the mutational genotypes of the population. So we're going to have a little bit of notation here. There's going to be this vector psi, and that's a vector that has the genotype of the normal, the reference, and the variant population, right? GN, GR, GV. So these would be things like AA for the normal, AB for a heterozygous mutation in the variant population. There's CG, and this is just a function that takes a genotype and tells you the copy number. So if it's AA, 2, AAA, 3, so forth. Then there's mu G. This is a little bit more tricky, but it's roughly, so what it is interpreted is as the probability of seeing a read with the mutation given the genotype. Roughly speaking, it's the number of chromosomes that have the mutation in the genotype divided by the total number. So if it's AB, 1/2, ABB, maybe 2/3, and so forth. Okay? Does that sort of make sense? Sorry, question? No? Okay.
Is it not always 0 for the reference and normal?
We'll come back to this. That's why it's not strictly, that's why I'm saying it's subtle. But let's go to the first part of this and say, what is the probability a fragment came from one of the three populations, reference, normal, or variant, right? And to compute that probability, we're going to assume that it's proportional to how common that cell population was. But the subtle thing is we're also going to assume that it's proportional to the number of copies of the locus that cell population has. Okay? And that's what leads to these expressions. So the proportion of cancerous cells is 1 minus T. T is the tumor content here. And this is the copy number of the normal cell population. The proportion of cells in the reference population is T. That's the proportion of cancer cells, times 1 minus the cellular prevalence, right? Remember, cellular prevalence is defined to be proportion of cancer cells with the mutation. So 1 minus that is proportion of cancer cells without the mutation. Then we have, for the variant population, tumor content times cellular prevalence, right? Okay, so there's an important, maybe non-obvious point here. Why do we include the copy number in these probabilities? So we have this infinite pool. Why is the probability we sample or read not just proportional to how common the cells were, the cells that we put in the blender?
Because if we have a small population but very high copy number, we're going to have more
Yeah, so that's exactly it. So the answer is if you have a very high copy number, the chance that you put a read into the pool is much higher. So the thought experiment I always do, what if you had one of the populations that had an infinite number of copies of this, right? Then it would contribute infinitely many reads to the pool and everything else would be finite. So we'd only ever sample reads from that cell population, right? So we need to include the total copy number of the locus in the model. Everything clear so far? Clearish. All right. So we can formalize this a little bit and say, let E be the indicator variable on which population the read came from, where that's in normal, reference, variant. Then the probabilities are equal to this, and this Z is just a normalization, so this everything sums to 1. Okay? Now, the question is, how do we compute the probability, given that we know the population the read came from, how do we compute the probability that read has the mutation? And basically, it's the number of mutant chromosomes over the total number, right? That's almost right. It's a little bit more subtle, though. Here, I'm just introducing one more piece of notation, B, and that's just the function that counts how many chromosomes have the mutation, right? So if you never have the mutation, if the cell population doesn't have any chromosomes with the mutation, does that mean we will never ever see any reads with the variant? Is our data perfect?
No.
Bang, right? So what is epsilon here? Sequence error. So why don't we have it when, if we have between 1, this should read CG minus 1, not CG. If not all of the chromosomes are mutated, then we get this expression, which is the one that we sort of think is intuitive, right? But why doesn't epsilon appear here? To understand that, maybe I should say I am making one other assumption, which is that sequence error with equal probability mutates you from the reference to the variant and from the variant back to the reference, right? In that case, there's an equal chance we flip one of them back and forth, right? So things basically cancel out if we actually have a chromosome with the variant or a chromosome with the reference. If all of the chromosomes have the mutation here, then we go 1 minus epsilon, right? So these are our probabilities. That may have seemed a bit confusing. Basically, it should be the number of mutant chromosomes divided by the number of total, but we have to modify that. When we have no mutant chromosomes, there's still some chance we see variant reads because of sequence error. And when we have all mutant chromosomes, there's some chance we see reads with the reference allele because of sequence error. That's all we're catching here, okay? So we can put that all together and compute the probability that the read has the mutation. In this notation, that's F equals 1. And here we're going to assume that we know the cellular, the mutational genotype, the cellular prevalence, and the tumor content. Then we apply the law of total probability, which, if you expand it out and write out each of these, gives you this equation, which is the probability of seeing a variant read given all these other things, right? In practice, we don't observe one read. We observe D of them, or sequence depth. Then the probability that B of those are mutants is then going to be binomially distributed with parameters d for the depth, and probability of success given by that same probability we just derived, right? Okay. Quickly recap, we worked through all of that to answer a very simple question. What is the probability of seeing a variant given the genotype, tumor content, and cellular prevalence, right? We've been treating those quantities as known. We don't typically know any of them. We're not going to worry so much about the tumor content because we're going to assume we can get a good estimate of that from some outside source. We're not going to worry at this point about the cellular prevalence because that's the thing we want to learn about. But we have this problem that we need to know the genotypes of those three populations to be able to evaluate that quantity. That's not something we typically know. In fact, we never know that. But we need it, and it's a nuisance because without it, we can't evaluate our probabilities. The idea here, and this is a standard approach in probabilistic modeling, is then to introduce a latent variable, a hidden variable that is this genotype. To do that, we need to specify a prior distribution because we've just introduced a variable into a Bayesian model, so it needs a prior, right? Question is, what prior should we use for that genotype, right? So remember, the genotype is this vector with 3 entries, the genotype of each of the populations. It's a discrete quantity. One prior could be sort of a uniform prior where we assume all possible mutational genotypes are equally likely. Maybe we have to cap the total number of copies at some level so that it's actually finite. Is that a good idea? Pardon me?
It doesn't look like it's like you have so many possibilities.
Right. It's going to be computationally hard because we're going to have to sum over, either we're going to have to learn them or we're going to have to sum over all of them, so it's bad computationally. It's also very uninformative. So we'll see later on that the mutational genotype has a big impact on the certainty of our estimates of cellular prevalence. So we need to do something smart here. We need to have a better prior. This is a case where we can't be lazy Bayesians and say we can use the uninformative prior. We actually have to have an informative prior. So we need to go through a process called eliciting a prior. We need to think about what is our prior belief about the genotype of a mutation. And again, here we're thinking about a specific mutation. And now I'm going to throw in some side information. If we do whole genome sequencing, we'll often have copy number profiles, right? So remember, if you haven't seen these before, this is a very cartoonish version. And we have 2 lines, the red and the blue. The red is what I'll call the major copy. The blue is the minor. You could think of them as paternal-maternal, but we just can't label them that way. So we just say which one's the bigger of the 2 numbers. It's the major. So here, for example, in this region of the genome, we have 1 copy of both. So that's a heterozygous, that's a diploid position. In this region, we have 1 copy of the minor and 2 copies of the major, right? So that's a total copy 3 region. So what could have caused that? Well, we would have had amplification of one of the alleles, right? That duplication of 1 chunk of there. Further on, we also have 2 copies of the major and 0 of the minor, so then maybe we had a subsequent deletion, right? And so this is moving along genomic positions here. So our mutation is going to fall in one of these regions. And if we have the copy number profile, we then know what the overlapping chromosomal copy numbers are, right? The major minor copy number, right? We can use this information because if the total copy number is 3, and then we can pretty much eliminate any solution where the variant population has 100 copies or 10 copies or 5, right? It should have 3 if that's what we know, right? So one thing we could do is we could enumerate all solutions where the variant population had 3 copies, right? I think you can do better. This is just a matter of personal belief and how I'm going to use that data to get to a prior. So I have a simple model that I use to take that information, where I know I have 2 major copies and 1 minor overlapping the mutation. And my model is to say, I'll think about 2 cases when I'm trying to assign my different situations. The first case is that mutation happens before any copy number change occurs, okay? And that's going to be covered by these 2 cases here, right? In this case, we have in the first one here, we only have 1 copy of the mutant in the variant population. That corresponds to the case where the minor allele is the mutant one, right? In the second case, we have 2 copies. That corresponds to the case where the major allele is the mutated one. Remember, I'm assuming the mutation happens before the copy number change, so it will then be amplified when we have the copy gain. There's one other case I consider, which is that the mutation happens after the copy number change. You'll notice here now that the reference population has 3 copies because basically all the copy changes happen, then we get a mutant. We will only ever have 1 allele mutated though, because the mutation is after the copy change and we're assuming mutations don't happen twice at the same position. This is my way of using that information to come up with a prior. You can think of other ways to do it. But then the question is, how do I weight these different scenarios? And for me, I have no preference to any of them, so I just give them all equal weight. Does this sort of make sense, or is this a little bit confusing? Okay.
But this copy number profile from the previous slide, is it for the whole T population of cancer cells?
Yeah, so this is the approximation. We'll assume that it's sort of somehow representative of all the cancer cells. And there's,
All the cancer cells.
if we have subclonal copy number information, this picture gets uglier. So we make the simplification that copy number changes are clonal. Another simplifying assumption buried in there. I think I mentioned it in the notes, so we don't forget it. All right. Till now we're clear. Okay. We can put this all together now and finish off the model. We'll need a prior for our cellular prevalence. That's the proportion of cancer cells with the mutation. If we have no other knowledge, we can just, this is one case where it's probably okay to be lazy and just say it can be anything between 0 and 1. I don't really know. If you do have prior knowledge, by all means put it in, but you typically don't for an arbitrary mutation in the genome. We're going to assume, as I said before, that the tumor content is fixed. You can relax this, but often we have a pretty good estimate from copy number variation analysis that tends to actually be more robust than what you would get from trying to estimate it out of this model. So it's better to keep it fixed, but you can relax that. Now we can compute the joint distribution, and once we have the joint distribution, the posterior. So now we put it all together. We write down the probability of our data, the data and all our model parameters, the joint distribution here. And we turn the Bayesian crank and compute the posterior, right? There's one wrinkle. This is not a closed form integral. So this is I've mentioned this in the first set of talks, it's often hard to compute the normalization if it doesn't have a closed form. However, this is a one-dimensional integral over 0, 1. That is incredibly easy to compute numerically. Plug it into any piece of software and it'll spit out this number. Then you can just evaluate these points along a grid and plot your posteriors, right? This is one of the cases where you can actually get that even if you don't have something like conjugacy. It's not super common, but it is actually often, if you do have 1D integrals, it is useful to remember that you can just do quadrature. So just plug it into some Python function that integrates a function. So here, it's killing me. We have our posterior distributions for cellular prevalence, right? So this is a case where we have 100 reads with the reference, 100 with the mutation, a VAF of what? 1/2, right. And we're going to assume, sorry, this is a typo. This should actually read copy number 2, 0, so major copy 2, minor copy 0. So in that case, we have essentially 2 scenarios. I'm not going to draw the normal population because it's always, unless you're on a sex chromosome in a male, it's always 2. But I will draw reference variant. So in the case of copy number 2, 0, my sort of prior looks like this. We can argue a little bit about this. There's a slight subtlety there, but that's basically the prior we're using. We have 2 equally weighted solutions that we consider. So when we look at the posterior distribution of our cellular prevalence then, we see 2 peaks, okay? Peak 1 is when we have this solution. Right? Our VAF is 1/2. If both of them were mutated, that would mean the cellular prevalence would have to be 1/2, right? The second peak at 1 is this answer. Our VAF is 1/2. That means the cellular prevalence is 1, because only half of the reads from that population would show the mutation. If I got that backwards, correct me. That makes sense. I see a lot of confused faces, but, or at least maybe just deep thought.
So 1/2 is referring to what ratio is it again? Is it understood right?
Pardon me?
The 1/2 on the side, what is it?
Oh, this is the prior probability. This is just the mixed weight.
You don't get this from the both, you get this from
You get what?
You get this from the both out of the left, or is it like?
No, sorry. This 1/2 is this number back here. It's just the, and it's this term here. It's the prior weight I give it. So this actually just goes back all the way to that picture I showed of the homogeneous population of diploid cells, and if they all were at cellular prevalence 1, we'd see VAF 1/2, right? That's this case. That's the posterior, and this one is the other peak, right? And then if we have different copy number profiles, we're going to have different peaks, right? I think it's lunchtime, which is actually perfect. 12:30, right? Oh, no, we still have an hour? Oh, good. That's even better. All right, good. So there's still half this module left, so that's good. All right, so then the key point here is now we can compute the posteriors of cellular prevalence, but let's say that when I showed this to people who are actually more biologically motivated, they weren't super happy with this, because how certain are we about the cellular prevalence here? I'm not very certain, right? I have 2 big modes, and it gets worse if copy number goes up, right? So at some point, we'd like to get rid of this issue, and that's what we're going to come to in a second. But just to reiterate, the reason we have 2 modes is because we have uncertainty about our mutational genotypes, right? That's why we have multimodality. Okay. So that sort of leads us into the next part of the talk, which is about mutational clustering. So far, we have a model that relates cellular prevalence to allele count data. And we've basically been imagining that model 1 mutation at a time. So we've been treating mutations as though they were completely independent. The downside of doing this, though it's simple, is we end up with posteriors that are multimodal, reflecting uncertainty in mutational genotype. How do we get rid of multimodality? And excuse the typo. You guys have read the paper?
Have FMC like at different points in the change, and you have like the couple change.
Won't save you.
1 second.
Nope, this is, that multimodality in the posterior and sampling it, you can get away from that by doing MCMC and stuff. This isn't an MCMC problem. We have the perfect posterior. It is truly multimodal. The question is, how do we actually try to resolve our uncertainty about mutational genotype and pick one of those modes? So it's not that our sampling is working badly, it's that we truly have honest uncertainty based on the data. And so the question, then, how do we get rid of that?
So when you say uncertainty about the data, is it because the modes are identifiable, or is it?
Problem is so thus far in the independent case, yes, it's unidentifiable because of mutational genotype. That is why the posterior is multimodal. That's the symptom of unidentifiability. As you see in Bayesian settings, unidentifiability is not a huge issue, you just end up with 2 modes, right? So you have it. But if you want 1 mode, then it's a problem. So if you want to say, what is the cellular prevalence of this mutation? That's what we're going to ask her. I don't want to guess it's one of these 2 modes. I want you to tell me which of those 2. How do we get there? Of that's actually what we are doing. That's why we're getting the multimodality. Without any extra information about mutational genotypes, it won't go away. That's actually what we are doing. So we could pick one of the genotypes at random, and I'd say, just forget this case. I only ever consider this one, and then the multimodality goes away, but that's kind of dumb because I don't actually have any reason to do that. The answer was in the name of the section. Bang. Yes, you don't treat the data as independent anymore. You look at all of the mutations jointly. This is the idea of sharing statistical strength that I mentioned in the first module. And it basically means that if we look at everything together, we can learn more than if we treat everything independently, right? Okay, why? In this particular problem, does that make sense? Well, we know somewhere in the background of all of this, there is an evolutionary tree that relates our clonal cell populations, right? A very simple cartoon of this evolutionary tree is on this side, where stars are mutations, and each node is a different population, right? As we move down the tree, we acquire mutations. those get passed down to the descendants. When we sequence, or sorry, the implication of this is that mutations higher up the tree are at higher cellular prevalence. They're more common. So, for example, a mutation in the root node isn't in all the cells, right? It's propagated to all of the descendants. To get to our question, though, the interesting mutations are the blue, the purple, and the green. They all occur or originate at the same point in the evolutionary history, so they're all propagated to the same descendants. And so their cellular prevalence should be equal, yeah, the same, right? So if we knew the cellular prevalences, we could actually identify which mutations originated at the same point in the evolutionary tree. Conversely, if we knew which mutations originated at the same point, we could group them all together to compute our posterior and share statistical strength among the mutations to infer the cellular prevalence more accurately. Okay? This obviously raises a bit of a chicken and an egg problem, that we don't know the cellular prevalence and we don't know the grouping. If we knew one, we could do better at the other. And this is a really common problem in probabilistic modeling. It's basically you have some clustering of your data, some hidden group structure, right? And the tool that we usually reach for when we have this is a mixture model. And the basic idea of a mixture model is to associate to each of our data points some hidden quantity, some latent variable, which is the cluster indicator. And this is just a thing that says, this data point, this mutation belongs to this group, clone, right? Once you know which group you belong to, then you know what the parameter used to generate that data. You know what the cellular prevalence, for example, of the mutation is once you know the group you're in. So I think this might be our first example of a hierarchical Bayesian model. And this is sort of the generic version for a mixture model. So this isn't any specific mixture model. This is totally arbitrary. And this is a Bayesian mixture model. And what it says is we have some prior kappa, and we sample these parameters rho from a Dirichlet distribution. So a Dirichlet distribution is a distribution over vectors of some length. Here it's length k. And the key thing is the elements of rho, if you sum them up, equal 1. It's the simplex in mathematics, yeah. So it's a distribution on the simplex, the k-dimensional simplex. Once you have rho, that acts as the parameter for Zn, and the distribution we assume is a categorical. So a categorical distribution is just a distribution where you draw one of some number of values, categories, right? And the probability of drawing any specific value is given by rho. That gives us our cluster indicator, our label where the data point came from. Each cluster needs to have a parameter associated with it. That's sort of what makes it unique. Here that would be, well, we'll see later, that will be cellular prevalence for our model. But it's sort of arbitrary. So that's why we have just G. It's some blank distribution that we need to fill in if we want to build a specific mixture model. Then the conditional distribution of our data, given the cluster indicator and the set of the vector of cluster parameters, is given by some other arbitrary distribution where now we've selected the parameter from the cluster we belong to generate the data. This is sort of a very abstract but general construction of a mixture model. So with all that said, we can turn the model we built thus far into a mixture model. At this point, we're going to assume we know the number of clones, so the K in the last thing, the number of components of the mixture. So this is a finite mixture model. Later, we'll come back to how do we learn that. But it just, things are much easier if you assume that you know the number of clusters in advance and it's fixed. So the nice thing is, and just to remind you from the earlier picture, is each cluster, what does it represent in this model? It's going to be a group of mutations which originate at the same point in the evolutionary history, the same point in the tree, right? What do the parameters associated with the clusters represent? The cellular prevalence of all of the mutations within that cluster, right? So now we have sharing of statistical strength between mutations within clusters. And we see we can kind of take that generic template and we modify it. So now we have to assume the prior distribution for the cluster parameters. Again, we'll just use a uniform since we have no real reason to prefer anything. And now we have a little bit of extra notation. We picked up this subscript n, which indexes the mutations, right? So we have bn, the reference counts, dn, the depth, and then the prior, the mutation-specific prior. So as we move along, each mutation will have different copy number, and so we'll have to come up with a separate picture like this for each of the mutations, right? Is that clear? Or? So recall, so pi here is just these numbers. It's 1/2, 1/2. Psi is just this picture, the genotypes, which we need to compute this thing. We can draw this picture because we had major copy 2, minor 0. If I move to a different mutation that has major copy 3, minor copy 1, I'm going to have a separate picture and a different set of weights, right? So that's what's going on here. But essentially now, all we've done is reuse all the work we did with the mutational genotype model and just embedded it right into this mixture model. So that's this distribution here. And now the only thing is that the cellular prevalences will be shared between different mutations. I'm going to stop before I talk about inference. Is there any questions on this? It's a little bit confusing, simple. All right.
All those simplifications, do you assume that if in the same subclone we have some group of mutations, and those mutations from one subclone, they also share the copy number?
We don't assume that, no.
We don't.
That's why pi is indexed by mutation. Later, when we go to multi-samples, we even try to relax a little bit more in a way I'll discuss. But no, that is not a simplifying assumption. Okay. If you go home, read the notes, look over the slides, and you have questions about this, email me. I think now things are starting to get a little bit more opaque. So the question here, okay, we're going through our steps, right? We've now built a model. How do we fit this model? In the last bit where we had the mutational genotype model, we could actually use numerical computations to get the normalization constant. Things were really easy. We've now moved to a model that is complex enough that's not possible. So now we have to fall back on one of the approximation schemes we could use. One of them is to use map estimation via the expectation maximization algorithm. And so if you have mixture models, this is a really common approach. We're not going to do that here. I'm not going to discuss that. We'll get to map and EM in the next module, but Here, ultimately, we are interested in some measure of uncertainty, so that sort of rules out the map estimate right away. It also turns out that when we go to the full model where we try to infer the parameters, we can't even do map estimation. So we're gonna end up at MCMC anyways. So we're just gonna go straight to that here. And I won't go through the details of all of them, but essentially, we just need to think about how we update each of the parameters. We're not gonna update them all at once together. We're gonna use a blocking strategy here. So here, the blocks are, we're gonna do a Metropolis Hastings move to update the cluster parameters, the cellular prevalences, and we're gonna update for each cluster separately. We're not gonna do them all together either. We're gonna use a Gibbs sampler to update the cluster indicators, and I'll show how that's done on the next slide. And we can use either a Gibbs or a Metropolis Hastings move to update the mixture weights. It turns out because the Bayesian mixture model has a conjugacy structure, it's actually pretty easy to implement the Gibbs sampler, and that's the standard approach. Again, I won't give you any details. Go look up mixture models. That'll be one of the first things that pops up. The one thing that is useful is to understand how do you do Gibbs sampling for the cluster indicators. To derive the Gibbs sampler, it's in the notes. Essentially what you need to do is compute the conditional distribution of the cluster indicator for mutation n, given everything else is held fixed in the model. So that's what this little dash means. It just means fix everything else, right? If you go through the math, it's a lot of things cancel, and it's not actually terribly complex. You end up coming out with this equation, and essentially every mixture model has this sort of form where it's the mixture weight, we call this, the kth component of rho, multiplied by the likelihood if the data point was a member of that cluster, right? And then normalized over all possible clusters. This is just a discrete distribution, so once you have those probabilities, it's easy to sample from. Just plug it into any sort of library in R or Python. But now we have, is that sort of clear thus far? So now we have a question. Right now we can freeze K, and we have a full end-to-end solution for this problem, or at least as far as building the inference scheme, right? But we don't know K. We don't know the number of clones, right? We have no idea what's in our sample before we look at the data. So the question is, how do we set the number of clusters, right? Has anyone ever worked on this type of problem before, has some nets? There's more than one way to do this.
What was the question?
How do we pick the number of clusters?
So, I mean, there are models that are assume the fixed number of clusters. It's like Chinese restaurant process or Indian buffet, that the number of clusters is have a parameter for going to the cluster, and then you have a parameter to create a new cluster.
Yeah, so that's right. So one solution is to build a more complex model that in some sense includes the number of clusters as an additional random variable. That's essentially what we will do with the Dirichlet process in the next few slides. But that's not the only way to do this. Here we've used MCMC. If you do map estimation in EM, which sometimes you do need to do for computational efficiency, building a Dirichlet process into that isn't really possible. So there's an alternative way to do model selection, And it essentially comes down to looking at some penalized form of your likelihood, right? So the common things that you will see in the literature are the Bayesian information criterion, the AIC, and I don't remember what the A stands for in it. Okay. Those sort of things. So it's basically fit your model, evaluate the likelihood, add a penalty, and then see which version. So we have to fit the model with a range of different range of cluster numbers then, right? And the penalty there is really about not rewarding overly complex models. So one issue with mixture models, if you don't do the fully Bayesian thing, especially if you do frequentist maximum likelihood estimators and you just look at the likelihood, it gets higher and higher the more components you add, up until you have as many clusters as data points. Because it can always fit the data better, the likelihood can always be higher at least if each data point gets its own value of the cellular prevalence or the cluster parameter. Right? So that's why you need the penalty.
Because it's an imposed product and not conventional. If you tend to increase the number of parameters, it will always increase the likelihood every time. So it tends to.
Yeah, I mean this is just a general problem with likelihood, maximum likelihood. If you compare models of different complexity, it always rewards the most complex one, or typically. The Bayesian problem sidesteps this because you have an additional term, which is the prior. And the prior tends to say, I don't want to put all the mass on a very complex solution. I actually favor a simpler solution. Okay, so there's a bit of an aside there.
But in a way, you still end up, because you still have to choose the prior, so you kind of not completely out. Is it like this?
No, you don't necessarily. And I'll talk about that in a second, but you get out of it more than if you had to do, you get some flexibility. Let's say it that way. Remind me of that when we get to the full Dirichlet process. So this is where things will start to get a little bit more complex. Before I introduce we are going to take the strategy of adding, essentially adding the number of clusters into our model and then trying to infer it that way. So sort of in a very roundabout way. Before we do that and introduce the Dirichlet process, we're going to take a different view on mixture models. And we're going to move away from this classical way that it's presented, where we always think of the cluster indicators, right? So remember, in a mixture model, if the cluster indicator of two data points is the same, what that really means is they share the same parameter that generated them, right? They're in the same cluster, they have the same cluster parameter. So that means instead of explicitly having these cluster indicators, what we could instead do is for each data point sample a parameter to stick into that F distribution. So each data point samples a cellular prevalence. Then we say that for example, two mutations are in the same cluster if their cellular prevalence is matched. So it's just sort of a subtly different way of thinking of the problem. The key thing here is if we just sample our cluster parameters, our cellular prevalences from an arbitrary continuous distribution, they will always be different and we will never get clustering, right? What a mixture model is actually doing is in the background, it's constructing some discrete distribution G that looks like this. It's basically, it places the weight on a specific parameter. Because it is a discrete distribution, there is now a non-zero probability that when we sample from it, we will get the same value twice. Thus we get clustering, right? This is a bit of a weird way to do it, but I think this is probably the most intuitive way to understand the Dirichlet process mixture modeling. So all we've done is we've said we no longer have cluster indicators. We're now going to sample a separate each mutation samples its cellular prevalence, and if they're the same, we say they're in the same cluster. In order to make sure that we actually will get things that are the same, we have to have a discrete distribution. For those who aren't familiar with probability theory, the basic thing that we're saying here, if you have a continuous distribution, this is basically the key result, the probability of ever seeing the exact same thing twice is 0. It's a bit of a, it's a bit of an abstract, but you have an infinite, uncountably infinite, like, let's just say infinitely many things. You're not going to see the same thing if you make 2 random draws, right? Whereas if you only have k things you can pick from, there's a chance you're going to pick the same thing twice. That's the basic result. All right. So that moves us to the Dirichlet process prior. So the Dirichlet process is what's termed a non-parametric Bayesian prior. You'll see people often say that's a bit of a misnomer. It's not that there's no parameters, there's actually infinitely many parameters. In a sense that I can make exact in a bit. Formally, it's a distribution over distributions, or a stochastic process. So again, in probability that you've probably dealt with to date, your random variables usually take values in, say, the reals, like 0, 1, or 0.1, or in the integers or something like that, right? So they're numbers of some sort, or they could be objects, they could be red, green, blue, right? But that's the type of random variable. You can actually put a random distribution over anything, and so we could sample from sort of arbitrary objects. If the arbitrary object we sample is itself a distribution, and this is a little bit abstract, but that's basically the idea, that's what a stochastic process is. And that's what the Dirichlet process is doing. It's giving us a random distribution back, all right? I'll come back to that idea in a bit more. There are 2 parameters in the Dirichlet process. There's alpha, which is the concentration parameter, is the term, and G0, the base measure. Alpha is the thing that roughly controls the number of clusters. It's not such a killer thing, though. So if you fix alpha to a certain value, yes, you're going to sort of dictate the model complexity. It will jump around, though. What you can do to sidestep that is put a prior on alpha that's vague and then infer it too. So you can kind of get around the model complexity enforced by alpha.
So then distribution you probably put a
Pardon me?
It's a constraint, but it's still like some concept.
Yeah, you put a vague gamma prior on it and it basically can float to anything based on the data. It's still a constraint. There's sort of a no free lunch here, which is if you assume nothing, it's a bit hard to build a model or control the complexity in any way. Typically, you kind of want alpha to be fairly small because you want clusters. You don't, if you assumed alpha was gigantic, all of the data points would end up in separate clusters, why would you do a mixture model then? So you tend to want to encourage it to be low. Anyways, that's the caveat. G0 is this base measure. What it basically does is it controls where the cluster centers are. So it's the distribution that says, what is my cellular prevalence going to be? So we pick it to be a uniform 0, 1, for example. Then our cluster centers are going to be real values that lie in between 0 and 1. If I picked it to be a normal distribution, they'd be real values that lie between minus infinity and infinity, so forth. The key property of a Dirichlet process is the random distribution that it generates is discrete. You can formalize that a little bit more by saying it's almost surely discrete, but it's discrete, right? That means if we go back to the last slide and we think about, okay, we need a discrete distribution to have any chance of clustering, we can use the distribution we get from the Dirichlet process for that, okay? It guarantees that if we then sample cellular prevalences from this, there is a non-zero chance that two of them will be the same and we get clustering. That was sort of the point of this whole exercise. Is that somewhat clear? So again, this does give us an idea of, so how is this useful for estimating the number of clusters? Well, in the finite case, we sort of implicitly construct this distribution that we're sampling the cellular prevalences from. That distribution will always have exactly k entries because we're assuming a finite model with k components. In contrast, when we sample from a Dirichlet process, the distribution we get back is random, okay? Here I'm being a bit loose with the actual technical details, but intuitively, the distribution you're going to get back is going to have a different k each time we make a draw from the Dirichlet process, or at least potentially. And as we do, say, Monte Carlo sampling or MCMC sampling, we're going to draw a bunch of these distributions with different k's, and that will approximate our posterior over k. And that's how we learn the value of k. Questions? So it's kind of the beauty of these hierarchical models. The change looks like nothing. Technical details are a bit of a beast, but the change is nothing. We now assume a base measure that's 0, 1. We assume a Dirichlet process generates our cellular prevalence distribution, G, right? Then we draw our cellular prevalences. Now we're drawing one cellular prevalence for each mutation instead of one per cluster like way back. And then given the data, the cellular prevalence, all this, we just plug it into the model. And that is, and it's sort of the basic version of PyClone, I think, which you guys looked at, right? Makes sense so far? I am going to not say anything about inference. It's in the notes. It's not that much harder than the finite case. You essentially, maybe I will say one thing here. Essentially, the only change is we don't have cluster indicators really. So we have to think about how to update that. The algorithm you end up with actually introduces cluster indicators as auxiliary variables, and then you end up with something that looks like the Gibbs sampling step I showed earlier, with one wrinkle, which is that you have to consider the chance you join a new cluster. So it's in the notes. But now I just want to move on to sort of the next step in our chain here. So we assume we have our method for doing MCMC inference. We now want to summarize the posterior that we've inferred. So remember that when we do MCMC, we'll have a set of samples called a trace. In our particular case, that'll be sort of a set of cellular prevalences associated with each mutation. Or actually under the hood, we may still encode the cluster indicators, which we can derive, right? The question now is, how do we report a best clustering, right? The problem, so the nice thing about the Dirichlet process is yes, we will sample a different number of clusters every time. problem is how do we summarize that information, right? You can plot a histogram, for example, over the number of clusters that you see, and then pick maybe the mode. Not great. Based on the discussion we've had thus far, the Bayesian answer is to introduce a loss function, okay? So the loss function we introduce here is something called the adjusted rand index, and what it measures is the similarity of two clusterings. So to actually make it a loss function, we're going to negate it somehow. But then what we want to do is optimize this loss function given our sample posteriors. It's a bit tricky because we have to optimize over this discrete set of all possible clusterings. There's essentially a heuristic algorithm given in a paper, and it's called MPAIR. It's cited in the PyClone paper, and I will add the citation to the notes at some point. But it's basically just a way to optimize this loss. The interesting thing about how it actually works is that it computes this thing called the posterior similarity matrix. So that's looking at our MC-MC trace. What proportion of the time do we see two mutations in the same cluster, right? So if we always see them in the same cluster, then we're pretty certain they belong together. Whereas if we never see them in the same cluster and the posterior similarity is 0, we're pretty certain they should be apart. This is a very useful way to summarize the data because it doesn't depend on the number of clusters, right? It just says how often are two data points together. It also doesn't matter how the clusters are labeled or anything like that. So we can use that to get a point estimate of the clustering, which is a nice feature. And actually, once we have a point estimate of the cluster, we can then compute a point estimate for the cellular prevalence as well, because we fixed the model complexity. We know K. We also know actually now which mutations belong in each cluster. And so you end up with something like this, your prior times your likelihood. Here we're taking the product over all of the mutations that are in cluster little k. You have to compute a normalization to do this. It's not closed form, quadrature again, it's one-dimensional. You could do something more complex, but again, quadrature is your friend here. And that's basically how you can get your inferred clusters and your point estimates of cellular prevalence. I should actually mention that this isn't in the original paper. This is something that only appeared in later versions of PyClone, and I don't think I've ever written down anywhere but here. But this is what's going on under the hood. So the MPAIR part is in the paper.
Just I understand this correct. So is this point estimate basically so it's not like Genomic where you have each class probability, or how does this look like? Versus,
Sorry, say that again?
Is this point estimate gives you a probability for each cluster, or is it like what?
No, it is a hard call. This mutation is in cluster 1, 2, 3, 4, etc.
And it gives you like, so I don't know, I'm do you say you start with 0 cluster, you add like a new cluster somehow, or do you have each?
No,
like its own
this is after we've run our MC-MC. We've looked at a huge number of samples, all with different numbers of clusters.
Okay, so each sample comes from a different
number of clusters, yeah.
Okay.
So there's a really naive way to get a point estimate out of that MC-MC trace, which is to look at, out of all your samples, which one had the highest probability. And that's actually pretty frequently done. The other dumb way to do it is just to take your last sample as the estimate. That's also frequently done. This is better, right? Because in some sense, we're using all of the information from all of the samples we've collected to say which clustering best sort of encapsulates that information.
So would you say it's like similar to consensus tree phylogenetics, when you have the SNV and you
Yeah, that's-
made a speed conversion. I mean, you have a certain speed for a tree, and you count the number of speed that are in the 50% of the.
Yeah, so, and that's roughly what the adjusted rand index as your loss function is catching. And so yes, it is very similar to a consensus tree in phylogenetics. They both are also very conceptually similar in that we're trying to summarize a posterior over a big discrete space, right?
Just a practical question.
Yeah.
How many mutations do you consider? I mean, do you consider all the mutations or maybe just
So here, well,
drivers?
no, you can do everything. I mean, it's just a scalability. It's just how much time you want to let the sampler run for. And that's actually a little bit more of an implementation detail than, so you can do it with all the mutations. In practice, the way it was, this model, this isn't quite PyClone, but basically what this was developed was deep sequence data from a targeted panel. There aren't drivers because we don't know what drivers are. They were just a panel of mutations we put in. So hundreds, though it can be run on thousands. I mean,
Was that the computational power needed?
it's not bad. You may need to use a better sampling scheme than just the basic Gibbs sampler. We have a paper on how to use something more advanced. I'll have a slide about it later. Or, like, to be honest with you, you just ditch the Dirichlet cluster process altogether and do variational inference, and you can scale it up to billions of mutations. So it's not really, scalability is not really an issue for the model. It's just an issue for the implementation.
Are you aware of any software that get rid of this assumption of infinite reads in the pool and do something like probabilistic, like sampling correction or sampling replacement?
I don't know if that's I don't think that would be the most important thing to change about the model, though. I think the more interesting extensions have been the ones that have looked at relaxing the shared mutational genotype of all the cells in the population. So the one that I know of off the top of my head is Bayesian. Peter Mueller's group uses another non-parametric prior to get this. I'm not sure how well it works in practice, but it's kind of a nice solution.
So, one more question on this. So when you have your point estimate for the number of clusters,
yeah.
I guess you also get some sort of, what I'm looking at is more like a probability for each, or like membership, sort of like probability over your cluster. So you have this cluster as like.
We don't have any probability. It's just a hard call. The reason we don't, too, is, and it would be hard to make sense of that, is that we don't actually know how many clusters we have. Like, you could fix K in advance, then you could compute probabilities potentially. That's not what this approach does. Maybe let's talk about it at lunch, though. We'll follow it up. I think we're getting a bit close to the end, but or after. Okay. So, last couple slides for this module. The most important thing probably in this problem to have any chance of doing it well is to use multiple samples. I will say single sample whole genome sequencing, this model will almost never work. Especially whole genome sequencing, deep sequencing, it may get a bit better. And this is borne out in the paper and the experiments. But as soon as you add a few samples, you can do much, much better. And the core problem is if you have two clones that are at similar cellular prevalence, this model can't see them in one sample, right? It has no information to separate them. But typically, if you have an extra sample, that's not going to happen again. They may just be absent or whatever, or just it's going to be a different mixture of cells, right? So you're going to have more information. And so the more samples you take, the better the more clonal populations you'll have a chance to resolve. In addition, you'll also do better actual spatial sampling of the tumor, right? One question that I've had in the past, is it better to sequence one sample deeply or do more samples at shallower depths? Answer is do more samples at shallower depths. Because in the extreme, that's single cell sequencing. Right. So it's kind of obvious, actually, if you think about it that way. Anyways, how do we modify the model to deal with this? Again, turns out it's actually pretty trivial, so it looks almost the same. The only thing we do is we replace the scalar cellular prevalence that we had, because we only had one sample, with a vector, where that vector has m values, where m is the number of samples. We sample that vector uniformly at random from a hypercube. So it's 2 samples, the square over 0. I was going to draw this. 2-dimensional hypercube over 0, 1 is this set. Just sample from the square. So you're going to have a vector where entries are between 0 and 1. Okay. And then you end up with some cluttering notation because you also have an index over samples. And then you also, the one thing that's a bit strange is you end up also with your genotype prior depending on the sample as well. And this is just the fact that different samples have different copy number profiles. This is the thing that goes a bit of the way to relaxing the assumption on the population structure. The other issue you run into with the basic model, you don't see this in the whole genome sequence data, but you do if you do higher coverage data typically, is that the data actually has way more variance than can be modeled by a binomial distribution. So binomial distribution really only has one parameter that controls both its mean and its variance. This is sort of a common issue you see again and again in model building. This is called over-dispersion. The answer is to replace the binomial with an over-dispersed version of it called the beta binomial. And then you basically set the mean to the same thing and have a free variance parameter to fit the data better. Right?
Is negative binomial?
No, beta binomial. Negative binomial is the over-dispersed version of a Poisson. We'll talk about that in the next module. But beta binomial is something different. It has support on the same support as a binomial, not over the integers. With that, we finish this module, I think in time for lunch. A little bit of time, actually. So that would be a good time for questions. Or I think you guys also have a tour planned after this. So. But are there any questions on this?
How would you say is the difference when you do like Bayesian inference in terms of the uncertainty estimates?
So I'll talk about that in the end of the module. If we did variational inference, the answer is typically it's worse. It's worse. I'll talk about that in the last module if we get to it. But
you still can get.
Yeah. So variational inference is somewhere between map and MCMC in accuracy, and also computational complexity.
And when does map not work? When can you not use it? Like, is there any
When does map inference not work? The reason it doesn't work for the Dirichlet process is because it's unclear how to optimize over a distribution over distributions. So there are maybe some ways around this using other approximations to the Dirichlet process, but that's the main issue, is that you can't really easily compute the optimum over this thing. All right. We good?
Sorry, but the way that you summarize the distributions even cooler or even better than consensus tree, because the consensus tree you could have a split that are not that produce a fully linear neighborhood. So this is even better, I guess.
Yeah, it is an easier problem because there's no additional constraints, right? So maybe I'm just going to say one last thing. I think we got a little too into the specifics of the model. I want to reiterate the process here, right? We started with the very general question about how do we take high throughput sequence data and get at clonal population structure. Once we looked at the data, we started to realize we have this whole issue of mutational genotype which confounds us. So rather than go for the full-fledged mixture model version at the beginning, we said let's do something simple. How do we even solve the mutational genotype problem, right? So we sort of come up with a simplified model of how we think the data is generated. We then build a model to correct for mutational genotype and associate and get cellular prevalence, right? Once we have that model, we see we have this issue with multimodal posteriors over the cellular prevalence. And that motivates us to then try to do something more complex, which is to use mixture models, right? But almost for free, we can just plug in all the work we did on constructing the mutational genotype model into our more complex model, right? Doing that lets us share statistical strength and resolve the multimodality. I didn't unfortunately show you a plot, but it does fix the issue actually really, really well. And then again, we have another layer of complexity because we no longer know the number of clones that we have, so we extend the model. And so as we work through, we have to think about how are we fitting this model. So sort of to keep things simple, I give you a fairly high-level description, but here we use Monte Carlo methods. That raises the question of then how do we report the posterior back? So there's this whole idea of the loss function where we optimize some criteria and give this optimal clustering in some sense, an optimal estimate of cellular prevalence. And then the only thing I didn't mention here is model validation. That's kind of not interesting. It's in the paper if you're curious, but I mean, it is interesting that we had to be somewhat clever about it, but it's maybe it's not very general. How we did it won't necessarily tell you guys how to do it for your own models, right? Whereas everything else here, the whole process is very generic. And then my final thought, people often think the interesting thing here is the Dirichlet process, the stat and the other clustering. That's the least interesting thing we did. The best part of this is the mutational genotype correction and the understanding of the underlying problem. It may be flawed, but it lays bare the assumption we made. And I think that's the more important part that gets overlooked, right? And that's actually, and we'll see when we do copy number, basically the same thinking comes up time and time again. Okay, let's take lunch. Yeah. Well, we still got more. You don't.